It's not simply clicking a button and getting something. It's about more of a science of relevance. So today's challenge is that we need fast results. Everyone, can we get a quick results in 2 days, but not understanding what is the computational capability that it has. We need a strong visual focus. We just look at numbers, score, energy, but how about our chemist? Because chemist wants to see the structure. Chemists want to see which region of the structure is toxic. Then only they can make arrangements to synthesize or not. So simply predicting numbers, chemists won't understand what, where, what is the starting reagent, all those things. And everyone needs it to be easy. Sir, is the software is easy to use? So the easy means basically understanding. How far you understood the science. If it is understood, then it is easy. So you shouldn't confuse between black boxing. Sir, A is easy, B is bad, very difficult. So I prefer using A, but you never concerned about whether A can give accurate results or not. So it is not about usability. It's about sound science of relevance. See, we need to understand, then things will be easy. Natural consequences, most of the time is algorithmic challenges. Whether A is good or B is good or C is good, we are always confused. A is the best in the world, nothing like that, right? So even you can use iPhone, even you can use Samsung, even you can use Me phones, all does the same job, calling a person, messaging, checking all your emails, social text. But what is the difference here? The cost and the branding, that's it. What is the accuracy there in the phones, right? But of course, quality is a very important factor. That depends upon the priority. So algorithmic challenges, until unless you try, how can you understand? So when you buy a smartphone, you ask 100 people to ask which is the best software, which is the best phone, or you might go to a shop and try yourself to buy one. But when you're choosing the softwares, you will ask one person, okay, which is the best software to buy or to use? He or she might request you based on their knowledge or based on what they have used for their project, but not necessarily the same target you're going to use for your project. Your target might be different. So algorithmic challenges are a bit different, because some algorithm is good for certain kinds of data or protein targets or molecules. Some other algorithm will be better for the other. So you have to try rather than just hearing. So you have to try yourself and then make conclusions. Non-specialists more involved. When you want to communicate structural information or a chemistry data from a computational modeler guy or a person to a synthetic chemist, the communication challenges are very trivial. Because modeler will always, this has a very good affinity, very good score, get me this structure synthesized. The synthesis person will say, no, this is very hard. It's so expensive with the reagents, so I cannot synthesize this. So the time for the modeler is wasted without something promises from the synthetic chemist. So these also should be materialized. And basic CAD skills are helpful throughout pharmaceutical R&D. Learning curve challenges are also there. As I said earlier, everybody wants it easy, but we also have to understand this is going for a drug, not for a game. So we have to put our efforts to learn the challenges. The common situation is that the chemist will mostly focus on the yield, synthesis, and generate a lot of data. The modeler is mostly concerned about the ADME, then pharmacophore and docking. But they don't talk to each other. I mean, they have communication challenges when there is a gap between the communication of both the experts. So we wanted to see a better solution where a chemist could understand what modeler talks and a modeler could understand what is a limitation of a chemist. So they are all being integrated. For example, a chemist could see the 3D molecular modeling results and can understand, okay, these are the regions where I could start making derivatives because there is some space left in the pocket. A modeler could also understand what is a synthetic feasibility, what is a retrosynthetic pathway for this particular newly designed molecule, so that a chemist will have all the details how to synthesize, right? So because you might be using SciFinder for retrosynthetic pathway predictor, right? The same way there are tools which could help you with us. And also a modeler could help able to tell which region of the compound is toxic, so that chemist will understand this is the region that I have to optimize. Otherwise, what we do as a medicinal chemist, we have A, B, C compounds. We give it for toxicity studies. We know that A is most active, biologically potent compound. And when the results of toxicity came, you came to know A is very toxic. Okay. As a chemist, how do you know where it is toxic? And an animal testing person or an expert or a toxicologist, whoever it is, they might not be able to tell you which region is more toxic. That is where a knowledge base is required to understand which region of the molecule is toxic. Then only a chemist can also optimize it. So these are things, communication challenges can be minimized by looking at understanding each other's limitations and merits. Now, how to do that? Of course, there are tools and how to overcome this. So that's where Thomas Lipinski said that a rational dialogue between medicinal chemists and computational models can be very, very productive. So you guys have to work together. In audience, I could say pharmacology and pharmaceutical chemistry, should work together. So when you're looking for a drug designing approach, based on the data availability is where what we have to look at, how things has to be taken care. If we have crystal structure of proteins available, I didn't mention homology modeling. Homology modeling and docking does not go well. If you are doing it, a lot of validation has to be done on homology modeled structures. When you have a 3D protein structures available, that is X-ray crystal structure available, that's where you do molecular docking. And certain softwares ask you to minimize, some does not ask you to minimize. All those things you have to take it. So in my YouTube channel, I have been discussing whether you have to minimize your protein or not. Literally, the answer is no. So it depends upon what software, what is the condition of the protein, whether you have to minimize or not. And ligands. In ligands, you have to make sure that you would have synthesized a compound, and that compound, when you crystallize, you get a geometry. When it is being consumed and it binds to a biological complex, it will have a different conformation. So the conformation of the compound as it is will be different than the conformation when it binds to the biological complex. The role of molecular dynamics and molecular docking is to find out which conformation binds to the biological complex. Most of the time we do docking and finish our paper saying that this is my inhibitor, but we never analyze whether that particular ligand is stable and is able to bind tightly within the pocket. That's where we require molecular dynamics. So we are not talking about 2001 or 2002, just with docking we get good papers. Now it is 2021. We need molecular dynamics to confirm molecular docking. Unless you do that, you don't get it in your papers in good journals. So that's where we try to do with structure-based drug designing, right? So it is not about how others did, it's about how the science is, and then we have to do it. There are certain restrictions in science. We have to overcome that. If others have done it, that is past. As I told you, the docking, whatever you have done in 2010 is not the same that you're doing in 2021. It is different, which I'll be discussing in a moment. The next study is that when you have 3D structures of protein, but you try docking with all possible ligands and nothing is docking, and then you might come up with a de novo or scaffold hopping based analysis. That is called fragment based drug design. The other one is when you don't have a protein available, no 3D protein, that means a crystal structure protein available or a validated homology model structure available, then you do just have ligands and its activity or its conformations. And that's where you try with pharmacophore or similarity studies and QSAR studies. So this is called ligand-based drug design. And when you don't have any of these data, and that's where you do high throughput screening or combinatorial drug designing, those things are also being done. If you see, the major portion of the research is happening on structure-based and ligand-based. That's where a major of them, and then comes fragment based and then combinatorial, right? Now, since you have not said nobody knows computational, so expecting that you know about molecular docking, which I'm not going to talk since you already said you're all working on computational. So I'll be just covering up what is this picture is about. So molecular docking is to determine the optimal binding structure of a ligand, where we are looking at a drug candidate or a small molecule, and we are trying to interact with the receptor, with the drug target, and we are trying to quantify the strength of the ligand receptor interactions. So ultimately, we are trying to see which is the compound and at what conformation it is trying to interact with the biological target. But we wanted to rank them, right? In order to rank them, we are quantifying the strength of the ligand receptor interactions. So let us answer a few questions. So if I'm explaining what is molecular docking to my grandmother, I would explain it this way. Where the ligand will bind in my target protein? How will it bind? How strong it could bind? Why it is binding? And what makes a ligand bind to the receptor better than the others? So this is what we are trying to answer from docking. Most of you might be thinking, okay, we just talk about scores. We just talk about what is a hydrogen bond. And finally, we just talk about what is amino acids being interacting. No, that is called old school docking. We want to come out of it. This is 2021, right? We want to discuss much more about it. So what first we wanted to understand is, we wanted to understand what's the catalytic function of that protein. We wanted to understand the key amino acids engaged in the mechanism of that pathway, which will be antagonist or agonist in your studies. So this is what we are looking at. The first, from the docking studies, you have to understand what is the catalytic function of the key amino acids of the protein. The second, if it's an enzyme, you have to understand the regulatory mechanism, whether it is we are targeting at an allosteric site or an active site. That's where we again understand whether it is antagonist or agonist. And the cleft detection. Simply predicting a binding pocket doesn't have a sense of biochemical aspects. Software will simply predict the pocket using geometrical parameters. That doesn't mean that the pocket is in the catalytic site. So you have to understand whether your pockets are in the catalytic site or not. The question you will say, answer you might be saying, I saw literature. There are many protein where there is no literature data available for catalytic site. So that's where we have to come up with the analysis. Then geometrical analysis. What is the change happens to the conformation when a ligand goes and binds them? Where there is a closing and opening of conformations happens. Then goes with flexibility, whether you are targeting your substrate bound or aposites. Conservation. If you're working on anti-cancer drugs and you're targeting a specific cancer cell, excuse me, how can you say that this particular compound is selective or sensitive to a specific cancer cell line or cancer drug target? So conservation studies are very important. So that's where selectivity, specificity, and sensitivity is very important. If you say that, yeah, my drug goes and binds all the target, then I found a multi-target. Might not be. It could be an off-target too. So that's where when you do this analysis, just targeting on a single protein doesn't mean that it is specific or selective. That you have to do a conservatory study. So you do a comparative analysis with related proteins to understand whether there is any off-target or multi-target options. Then surface characteristics. We wanted to look at solubility criteria, solvent accessible area, and many more other factors. That is very, very trivial and important. Just a minute. Yeah. So, and also the interaction energy. Interaction energy, basically, we are looking at how the van der Waals, then electrostatic energies, and other hydrogen bond, all factors come into the picture, right? Moving forward. So this is a picture that I've taken from a book called Enzyme Methodology. So ultimately, the output of docking, you're expecting to see what is that you're getting out from a docking, not just scores. Now, moving forward, what are the challenges? Ligand and receptors are conformationally flexible, but most of the time we make receptor more rigid. And keep in mind, hydrogen bonds are weak, short-range interactions, including salt bridges, hydrophobic contacts, electrostatics, van der Waals repulsion. And binding affinity is a difference of uncomplexed state, which is solvated and desolvated, and which plays a very important role. So score does not have importance when you're doing or finalizing your compound. Score has importance only when you are doing virtual screening. And docking has just two simple questions to answer. What's the pose of a ligand in a pocket? Getting in? How happy is the ligand that is scoring it? So basically, we wanted to visualize in the opt and a follow. The previous slides, which I skipped, was just a heading title. So that's why I skipped it. So initially, when you are loading a protein, you wanted to see protonation, not simply a protonation. There are more details to be done in protonation. That is called protein preparation step. You have to optimize and score them. You have to visualize them and you have to edit them. That's where we look at visual torsion capabilities and other. So that is very, very important how you take care of this. So ligand and protein preparation is where we are looking at tautomerism and also wrong element assignments when the isoelectric are taken care and zero rotation, right? Is there some questions? Oh yeah, there is. I look like something is there. Just a minute. Oh, okay. Yeah, there was a question. You have mentioned in your talk that protein minimization. Yeah, I think since I am in the right place, I think I'll explain that and move forward. Great. Yeah, so you can put your questions in the chat. Anyway, we will answer them at the end of the session. But since I just saw some pop up, I'll just cover that up. Okay. Just a minute. So kindly post your question. That gives a confidence that whether you have understood or not. I don't have an intention to complete all my slides. At least whatever being shared has to go properly. Just a minute. Yeah. So the pseudo rotations, we also have to understand the orthopara positions in which side, because this is very, very important. We wanted to see which portion or which side is facing towards the pocket. When you do protonation also, this is a very important factor. Adding hydrogens, we simply, very in a simplistic way we say, but adding hydrogen also has importance in orientation. Of course, the bond length and bond angle is taken care, but the question is the orientation. If it is facing towards left or right, backward or frontward is very, very important when you do protonation step, especially on the binding site. It takes a very few minutes or seconds, like milliseconds speed, high accuracy, and hydrogen network for every pose is affordable, and real interactivity is also now possible in docking studies. So how the conformations are taken care? So we try to split the compound in different fragments, and we try to generate conformations within the pocket to see which conformation doesn't duplicate or overlap with the proteins, which is very important because you shouldn't have any overlaps. So it is much faster and it is much quicker. And then we say what is affinity, binding energy. So when I'm saying unbound with water, you can say water is not bound, and this is the ligand. And here bound in the complex means there is water as well as ligand being bound. So we are trying to take a delta G, means delta G of unbound water and delta G of bound complex, and that's difference is what is delta G. That is what we call it as binding energy. And finally, about the solvation bill. So how far, so you can see here, this particular ligand is very unhappy to be taken out of water. But this situation, it's very happy. So don't think I'm talking about the water molecule present in the crystal structure. No, I'm talking about the water environment. So you solvate your system later. So the water might be also very unhappy, which to not bind water anymore. So both situations are possible. So we don't know which one might be happy. So for example, for our ligand, this might be unhappy, but this will be happy. In this situation, this is unhappy and this is also unhappy. So we have to see both the situation. That's why I told you in the previous slide. So we are taking a difference of both conditions, unbound and bound, right? And then we are looking at a different situation where when this gets inside, what I said a few minutes back. So it might be very unhappy to get locked into a pocket also, and it might be very happy to be freed out from the pocket, both situations. So we wanted to see both bound and unbound to come to a conclusion. Whether to understand whether this is a good or a bad affinity. Now you can see the hydrophobic part of the ligand is a bit exposed. The phenyl part was unhappy in water and still it is. So that is the situation I was telling you. Also, it does not form any new hydrogen bond or the like where there is no contribution. We expect it to have some contribution, but it did not have contribution. So that's what we are looking at, both aspects of solvated and desolvated. And delta G approximation, the partition coefficients like log P connects to the dehydration and hydrogen bond in a natural way. And computation is super fast and atomic increments. So we are looking at more of a visual affinity. So what is the binding affinity is more involved with? It looks at clashes and torsion energies. That's what we are looking at. So I'm sure if you are using Autodoc, you would have seen intraligand and interligand clashes. If clashes are there, please do not take those data because you have to increase the confirmations and come up with a confirmation or process where there is no clashes. It's very, very important. Apart from these factors, there are some other important factors like torsions. I told you earlier, you're doing and performing docking in order to understand whether the conformation that is bound to the biological complex is crystallizable or not. So that's where we look at the dihedrals and torsions. So we could try to check with the Cambridge structural database where it contains only X-ray crystal structures. And we could see that whether the particular dihedrals or torsions are present in those databases or not. It is a statistical comparison. And that gives you in relevance the torsions are taken care. And ultimately for a medicinal chemist, this is very important. It helps you to understand where to be optimized. So here, the red colored balls tells you this has a very negative contribution to binding affinity. Wherever you see a green ball, it tells you it has a positive contribution to the affinity. I didn't talk about scores anywhere. So when you make optimization as a chemist, you might think of making modifications. So here the tools and algorithms will be able to highlight which region of the compound is problematic so that your affinity is being affected. So you can see the energies here, desolvated energy and for the receptor as well as with the ligand, and the solvated energy for the interaction for the ligand and the receptor. The total of this is called 7.6 kilojoules per mole. If it is positive, of course, it will be red. If it is negative, it's more stable. You can see here it is minus 3.4 kilojoules per mole. So this is where I was telling you, we wanted to minimize the communication challenge between a chemist, organic chemist and a computational. So that here it tells to the chemist itself, this is having less affinity. So chemists can always make modifications here. So we can do different kinds of screening using any docking tool. And there we started with a virtual library of 45 lakhs. and we ended up with 172 compounds, and we had only 116 hits molecule. This doesn't mean I found a drug molecule. This does mean there are 116 molecules which has biological affinity. Let us see the difference of binding mode. This is where your score confuses you. For both, the scores will be good. So you can see here, there are two interactions happening. One on your left-hand side amino acid, one on the other on the right-hand side of the amino acids of the pocket. And both will have a different binding affinity, right? And both will have a very good score also. But so these are the two situations. Let us see what is a predicted binding affinity for both the situation. On the left, we got a minus 23 kilojoules per mole, and the other we got around minus 12 kilojoules per mole. So you could see that when the positions of the amino acids are changing on left and right, you could see the binding affinity is also changing. So this is what you have to look into it. So simply binding in the same pocket, it doesn't make sense. It has to go with the relevant amino acids also, and also the right conformation it is binding. So all these factors to be considered. So when you click a button and it is sorting based on the score, it doesn't mean all these things factors are involved. You have to have a visual focus. You have to look at each one of them and start analyzing them. So this is the other part which I was telling you about, the fragment based approach, where you can do core replacement. Let's imagine this blue part is the whole chemical compound, and you're replacing one single region with another fragment. That is called core replacement. Merging means, let's imagine you have a very big pocket. In one corner of the pocket, you have a small ligand. On the another corner, you have a very small ligand. You wanted to merge them. So on a chemistry terms, we could say conjugates or hybrids. So how you can link them, or you're finding the best linker. And then you have a small ligand, but the pocket is a bit huge. You know that this ligand is very promising, but you want to see how far it could be extended. So then a growing parameter also can be used. So this technology is being developed with by Roche, and also they use it for escape from patents. So when you replace this particular middle, the center region, so it escapes from the patented molecule. That is also possible. So how this is being done? We try to first generate a fragment creation. Then we define the query where we want to cut as a chemistry can synthesize and link the bond. So can you see here, I'm talking more about retrosynthetic shredding rules, where organic chemist knows much about it. Then a chemist can synthesize. A chemist knows where to add a substitution reaction, where to break a bond, all those things. So when a chemist sits and does this job, then that's where the synthetic accessibility will be also coming into the picture. Even the software can take care of most of the queries, but not necessarily everything, right? Any software, if someone is saying our software can do everything without a chemist, I don't believe that. It is not possible at all. So we need the intervention of a chemist expert to come up with fragment-based drug design more feasible and more meaningful. And finally, we link the forbid unwanted bond formations too. So let's say that we have different fragments extracted or prepared from drug molecules. By using different algorithms, we will be able to identify which are the key fragments which could come together. And we use our protein target to see which could be the best combination of the fragments, and it will form the full compound. I hope everybody knows what is Gleevec. Gleevec is the commercial name is imatinib, the anti-cancer drug. Now, if you ask me what is the success stories with the computer-aided drug designing, there are more than 112 compounds available. The first one with the Nimbus, yeah, it was expected to be successful, but it is nowhere going. I think they are still doing some optimizations, so we have to wait and see. But still, that's how it is going, right? And Nimbus, they're using all possible softwares worldwide. It's not just single software. They're using multiple softwares in In-Silico to come up with the best possible inhibitor. Similarly, many others, Bristol Myers, Merck, Gilead, and Novartis, and many more are working on computational studies. So the computational studies are giving assist. They do assist in the findings and making decisions. I've taken from these two reviews, basically, this list. Now, what is the objectives of CADD? This is where pharmacology comes into the picture. So far I was talking about a combination of chemistry and pharmacology. Now it's more of pharmacology, including genotoxicity and impurities, all those comes into the picture. So when you say I did docking and I did bioassay studies or binding affinity studies, you're just finding a hit molecule. You nowhere reached a lead or a drug molecule. There are three terminologies you have to learn in this particular slide. One is hit, another is lead, another is drug. Hit molecule is just a molecule, just we are finding out whether this is a promising inhibitor or an activator on a micro level. We already assume that it has reached the target, and we are seeing whether how the affinity and bioassay is there or potency is there. That's all we are looking at a hit molecule. In order to call the molecule to be a lead molecule, of course, you have to check the pharmacokinetic properties, including ADME properties, to see how far they are safe, that means toxicity, and what is the absorption rate, how far it is absorbed by the small intestine of human, metabolic stability, when the drug passes through different metabolic pathways, how far your drug is stable, right? For example, if you're looking at cytochrome P450, there are different isoforms like 3A4, 2DC, 2D9, 26, 2C6, 2C9, all those things. So you have to understand if it binds with any of these isoforms and the compound breaks down and form a metabolite. And in that case, it will not reach the target as desired because you found a hit molecule ensuring that your compound has already reached the target, drug target, right? And then you also look at solubility, aqueous solubility or flexibility, all those things factors also looked at. That's where you look at the lead or drug-like molecule. For me, or in general, in order to call a molecule to be a drug molecule, unless it passes clinical trials, we cannot call them a drug because it has not been ensured it's safe for humans. Lead molecule, drug molecule, we never tested. We claim being not tested on humans, but mostly on animals. So we have to identify chemical compounds with an optimal balance of all these properties is very trivial, and quickly identify such situation when such balance is not possible. And this slide is the answer for my first question in this session about how to reduce the cost of the medicine. If you are not able to find out a good balance of these properties, stop it. Stop with that molecule. Start working with other molecule or start trying to optimize them. So fail fast, fail cheap. If you are able to identify this is not going to work, stop it. But I know in academic setup, we might have already focused on a work plan. Once we start, if it doesn't work, still we try to somehow come up with the data and also try to satisfy whatever we have submitted. So that is where the beauty of research, it is trial and error. You might think of plan A, but later end up with plan B, C, and D. Of course, we all have done PhD. So I'm sure many of us have faced, I have faced, my plan A, plan B didn't work and plan C worked. Of course, during the middle, the committee, we reported that we are changing, the objective is same, but we are changing whatever we have reported earlier with the key compounds and other things. And that's highly acceptable because you propose something doesn't mean that it should work. We don't do magic, right? So that's how it is. So we have to be very sure we have to quickly identify that situation when such balance is not possible. Otherwise, that's the reason why many of the funding agencies are not able to give you funding, because they try to analyze a proposal where already a groundwork is already being done. That is what I was trying to tell you, because PhD, we don't do that. We do some more of literature work, but ultimately when we start doing it, we realize it. But when you're applying for funding, if you've done some groundwork already, some analysis, some wet lab studies, then the funding agency also will have a confidence that, okay, this guy has done something, so they know better what could be expected, what does not work. So it is worth funding them. So these are all simple tricks that we have to understand when you're applying for funding and all. And we have to be very confident. You move forward with a compound only when you're confident. If you're not confident, don't go. You're wasting your money and time. And avoid missed opportunities. As I told you earlier, we try to copycat some of the strategies used by some other people, but doesn't mean that that is going to work for your project. So we have to be very careful what strategy to be implemented and what kind of approaches to be implemented. So you have to avoid missed opportunities. I will cover this in a bit moment. So there is something called as chemical space, because earlier days when I was starting with my career, I used to do something. Pick a FDA approved drug of its confirmation, do a PubChem search or some database search, similarity search, pick up some compounds, and then try to do docking, QSAR, pharmacophore, and design some new molecules and ADME. But ultimately, we have to understand something, and which I stopped later. What we have to understand, this is called a chemical space. Any compound with yellow color is good. Any compound with bad is bad activity. I'm talking about the binding affinity and acid affinity. You could see here a compound here, yellow color, and you could see a compound which is red color. All the dots represents chemical structures, each one of them. Even though this compound is good, you could see a very neighboring next compound is also very bad, which is trying to tell here is, even though your structure is similar, 70%, 80%, 90%, need not be similar with your biological activity. So similar compounds might have physiochemical properties similar, not biological activity. So we have to be refrained from that. And if you come here also, there are many compounds which is active, but there are still one or two compounds not active. Here you can see there are many compounds clustered together. One is very active, the others are very bad. So this is what we have to understand when you're considering just a similarity for making decisions, not the right way. And finally, this is what I was telling you, how a chemist will know which region of the compound is toxic and how he can or she can optimize it. By seeing some representations like this is called glowing molecule, it will tell you which region of the molecule is problematic or which region of the molecule is toxic and which region of the molecule is non-toxic. So when a chemist sees that, okay, this is being from a knowledge base means from clinical trial, they have understood this particular region of the compound is toxic region. So that's how you get the knowledge base, not simply based on publications. Because don't think that all publications are accurate. There are many papers where there are mistakes. And when you write to authors, some they really don't reply. Some of them reply saying that was a mistake, this is the correct data. So that's what it matters. So don't think even Nature papers also, there are some mistakes already, and there are papers being retracted from Nature also. So don't think that looking at the impact factor, these journals are always accurate, because it all depends upon the reviewer. So if we miss, I'm also a reviewer to ACS, Willy, Elsevier journals, but doesn't mean that I don't make mistakes. I would have made mistakes also. So that is what we have to look at. Right. So finally, data overload is also, so it's time for another question. Hope you will be responding now. Let's imagine that you have 10 different molecules with you. You have to carry out 10 different experiments. Experiments means you have to understand which are the best 2 molecules I have to patent and pass it on to pharma industry to go with preclinical or clinical trials. So among the 10 experiments, all 10 experiments are mostly important or to be considered when you're ranking the 2 molecules. Now my question to all of you, and again only 50 seconds, you can either unmute or you can put it in the chat. You have to tell me how to consider all the 10 experiments data for all the 10 molecules to choose the best 2 molecules at the end of the day. Finally, also want to mention all these 10 experiments are independent because they are also important to make a selection of 2 molecules. So 15 seconds to go, you can start answering. I hope you can see. Yeah. So my question was how to choose or select the best 2 molecules from the 10 compounds from 10 different experimental data. I hope you know where to click on the chat and then paste it. So 15 seconds, 15, 14, 13, 12, 11, 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. Yeah, I got one response. Thank you so much for that. I don't know, we have more than 40 people involved in this session. So when we are not able to answer, how can we expect our students to respond? Anyway, just kidding. So one of the answer I received was statistics. Yes, it is statistics based. Most of the time, people used to tell me to take an average, and otherwise they will say, do one experiment by another experiment and filter them out. Both cases does not work when you're working on drug discovery projects. In drug discovery projects, we have a different approach, which I'm going to discuss. And thank you so much for the person who responded to this question. So we have a lot of data that is coming up, in silico data, in vitro data, in vivo data, right? How do we select a compound based on all these data together? We have to prioritize. Don't think that all the properties are important. A specific property might be important, not all the properties. So we have to also check which property is more trivial. This is very, very important. Some property will be more important than the other property. So that's where we have to look at it. So we have to give importance based on the quality of the data, based on the physical relevance to the activity or what kind of disease you are looking at. Another thing is uncertainty. As earlier I mentioned, don't think experimental data is accurate. It has its own drawbacks also. And don't think X-ray crystallography is most accurate method to get the structure elucidated. No, NMR is the best accurate method. But unfortunately, most of the proteins are not soluble, so we cannot use NMR. X-ray crystallography also changes from person to person. If I do X-ray crystallography and then I do model the protein, and if someone among this participant is also modeling the same one, we both will get a different data. So that's how it is, right? So there are uncertainty in experiment. When there is uncertainty in experiment, the best example I could give is titration when we were doing our schooling. So we were doing titration. Of course, we take 3 readings and also we report mean standard deviation. At that level, we learned it. But when we came to research, we forgot it. In experiment, we have to iterate our experiments. So we have to do it for 2 or 3 times and then talk about the standard deviation and mean. Most of the time we don't do because of the budget constraint, because repeat an experiment, it's again cost comes into the picture. So we cannot do. But then that single reading of your experimental data, let it be any rate data, it is uncertain, it is unreliable. There are a lot of errors involved and we don't know how to justify that. So when experimental data is having uncertainty, how do you think that predictions using software shouldn't have a standard deviation? So whatever you're predicting, whatever property you're predicting, there should be a standard deviation. If there's no standard deviation, personally, I don't take that data when it is predicted using software. Let it be any software. Finally, coming to selection, just clicking a button, ranking top 1, this is the first rank, this is my compound. That software set, but we didn't confirm it. When you're making selection, it should be manual. You have to consider both quality and diversity. That is very trivial. I'll tell you the importance of quality and diversity in a moment. So this is what, I didn't tell the answer for the question for 10 molecules and 10 experiments, which I'll be telling in a minute. But this was one of the scenario I was telling you most people used to tell this is how they wanted to do it. Let's imagine you have a set of molecules and you're doing potency studies or biological assays. Expecting one single compound is very active in that, especially the diamond shaped one. All others are stones. And we remove some of the compounds from there, which is inactive, right? And the other filtered one, we expect these are all very good active compounds. And finally, we did another 2 experiments on absorption rate. And again, we removed some of the molecules, expecting the goods going to the next. And metabolic stability, few bad molecules are being removed. So in each step, we expect that we are removing bad molecules. But keep in mind, we are removing bad molecules of that specific experiment. First for potency, second for absorption, third for metabolic stability. At the end, we expect that we are getting a very good compound. Unfortunately, more than 95%, this is not the case. Your good compound might end up here. Meaning, even though your compound is very good potent or active, the absorption rate of your compound might be less compared to the threshold that what you have considered, right? So if the absorption rate is less and your it has not met your threshold, of course, it will be out of your best list. So this is where optimization is a big factor that has to become in. We learn a lot about lead optimization. I have seen in many of the, when I go for academic council meetings, I used to suggest this. All the syllabus, there'll be lead optimization topic will be there, but very less importance given to that. In a pharmaceutical chemistry, pharmacology, the more importance to be given to lead optimization. That is the sole and only job for when you go to pharmaceutical industry. The rest of the things is a standard thing anybody can do, right? So that is the artwork that is, or creativity coming out from a human expertise about lead optimization. Anyway, so, and you end up with a non-promising compound. It's just because even though your compound is very potent, but does not have good absorption rate, it is out of the list. So you have to be very cautious when you're trying to do a filtering approach. That's what I exactly told you earlier about the chemical spaces. There are good active compounds, even though they are similar, there are both good and bad with closer greater points. And this is about uncertainty, which I already discussed. Single measurements, assay variability will be there. Multiple replicates where you understand the mean and the standard error or standard deviation. Statistical uncertainties in predictions are also there. So make sure that your predictions will have the standard error and standard deviation. That's the solution here is, I am sure that in M-Pharm pharmacology, I've seen in the syllabus, you have something called as multi-parameter optimization. So multi-parameter optimization is something similar to your picking up the properties. You also have desired value, but you give the level of importance. Which property is more important than the other? The good factor here is, let's imagine that you have a Lipinski rule of 5 where molecular weight is less than 500. Do you think a compound with molecular weight 501 is a bad compound or it doesn't work? But when you give a criteria like this, molecular weight less than 500, it will search only compound, it will consider only compound with less than 500. It will never consider compound with 501. And do you think 501 doesn't work? Never, no. There are FDA approved drugs which is more than 600 molecular weight. So on your normal way of strategy, you might miss compounds because 501 is also promising. Just think logically, it is promising. So why do you need to avoid them just because of a rule? So, but this importance will consider a 501 also, but with a lesser score or with a lesser importance. That's all. But we don't neglect them. So that is the difference between the previous slide, what I was neglecting compounds as it was being filtered out. So this is a very good example. At the bottom, I have given the citation where they took some Pfizer candidates and did analysis for CNS drugs, right? Central nervous system drugs. And when they were doing a normal filtration process, they found around 60% of the Pfizer candidates. And later, when they applied the multi-parameter optimization, they found additional 14%. So around 74% of the marketed CNS drugs they were able to identify, considering the top properties like log P, TPSA, log D, hydrogen bond donor, molecular weight, and PK. So that 14% they didn't find earlier, that is called the missed opportunities. So this is why I told you, if you're implementing wrong strategies or wrong rules, you might end up missing compounds from your I know many of us have got a good practice saying that First select a database, filter them with the Lipinski rule, and then do docking. No, don't do that. Let the database go. But when you do with phytochemicals or natural compounds, I do recommend, because phytochemicals are too rigid and too much molecular weight. If you know the size of your pocket, try to restrict the molecular weight and other things accordingly. That again, as I said, don't take these rules common to all kinds of data. It is very specific to data to data. That's where we also come to QSAR, because I am now entering into ADME. So before talking about ADME, that's where we wanted to know how to predict, or how the models are being generated to predict ADME for our properties or our products, structures. So you have a compound, it could be 3D or 2D. I am a personal lover of 2D. 3D has its own issue. I am not again discussing in my YouTube where I discuss a lot of scientific things. There I have discussed about why 3D QSAR has its own challenges and issues. We extract molecular descriptors and response variable, and we combine them, either regression or partially squares or support vector machines or random forests, or using machine learning modeling. We try to compare them. We generate a model, and then we validate it, come up with activity and property. And of course, for that compound, we need to have experimental assay data in order to generate the response variable. And at the end, you can see we could generate an equation for the prediction. At the end, I have plus or minus error. I have not seen many papers with this error given in the equation. They just write the equation, that's it. Error is not at all mentioned. This is what I was telling you. Error tells you the confidence, how far this model is reliable or not reliable. Nobody says if there is an error, it's bad. No way. Prediction always has error. That is a rule of statistics. So please ensure that when you're publishing QSAR related papers, standard deviation error is reported. Now, what is structure activity relationship? You get experimental data, activity mechanisms, and theoretically you have the structure, reactions, and chemistry, and empirically you have the method of analysis where the statistics. You combine all together, the activity, reaction, or the chemistry data, structure data, and the statistics. That's where SAR is formed. Structure, activity, relation. So structure is from chemistry, activity is from biology, and structure activity relationship is found from statistics or machine learning, right? And the same way, how to obtain a model. You have chemical molecules from biology and chemistry-based functional classifications. You extract the properties of the chemical structures, which is mechanistically relevant. That is very, very important. I know you use 100 softwares, 3,000 descriptors, 2,000 descriptors. What does you use if you don't know the meaning of the descriptor? That's what around 8 years back, I wrote a review on QSAR, which we clearly mentioned about it. Simply generating descriptors and R-square doesn't mean QSAR is good. No, you need to have mechanistically relevant descriptor. When you're referring a descriptor with an activity, there should be physical significance. Otherwise, no use. I'll tell you an example if there is time. Then quantitization, mechanistic characterization on biological activity, and you combine them with statistics, and that's where the SAR model comes in. And keep in mind, just getting better R-square doesn't mean it's a good QSAR. The better applicability domain talks about QSAR. And I've not seen many of the papers where they talk about applicability domain. There are regulatory uses of QSAR. So that's where it's very, very important. So simply generating QSAR doesn't mean value. You have to follow some regulatory use. And I'm sure there will be people from regulatory, you're learning regulatory things also. When you're doing QSAR, there's something called reach point. QSAR model relevant to regulatory purpose. OECD principles, that is scientifically valid QSAR model based on documentation. Another is QSAR model applicable to query chemical. Very, very important. That's where we talk about applicability domain, the scope of the model relevant substance of interest. If a software, any tool, online, offline, paid, free, whatever, if they are not able to talk about applicability domain, I don't rely those data. Of course, whatever I have published, I have already discussed about applicability domain. Without that, there is no value of that model. We don't have a confidence on understanding whether this model is reliable or not. So this is very, very trivial. And finally, this is a very standard format QMRF or QPRF. This is based on OECD principles. This is how this should be uploaded. To be honest with you, when you're publishing papers, if you're able to upload this as a supporting information, your paper will be very having a high value. So practice it. Many of the European team on QSAR, they always do this. In India, sorry, I have not seen much. They are talking about QMRF. And these are the free tools available, which generates it. Free as well as paid, both are available to generate this QSAR models. Regulatory means animal testing prohibited, because that is very important, right? We are in India also, we have from UGC Center for Alternatives, Animal Testing Alternative Centers are also available, where they use tools, softwares and others. So animal testing prohibited, cosmetics can use only these SAR models. In EU, use of alternatives to animal testing is promoted. In India also, there are 7 centers nationwide, and more are coming up. Chemical safety assessments, REACH, registration, evaluation, authorization, restriction of chemicals. PETA, people for the ethical treatment of animals. LCSA, Nuremberg Chemical Safety Act. IATA, integrated approaches to testing and assessment. TOX 21, which is adverse outcome pathways. So the regulatories are becoming more and more stronger. We have to learn this SAR, QSAR in the right way, not simply clicking a button and getting the data. These many tox properties are already available in EPA, Environmental Protection Agency from US. So all these can be predicted, and even developmental toxicology also can be predicted using QSAR models. Of course, check with the error. If the error is more, don't take that data. Doesn't mean that the software is bad. It just tells you that your compound chemical class is not present in the training set. When you're coming to ADME, we are talking about bioavailability, absorption, liver metabolism, permeability, lipophilicity, and hydrogen bonding. What is ADME? A is for absorption. We are trying to understand how the drug will get inside through the intestine. D is for distribution. We are understanding where will it go after being absorbed. Another is metabolism. Will it remain intact? Will my compound be very intact until it reaches the target, or it will break down? And E is for excretion. Will it stay in? Will it accumulate, or it go proper without any affecting the body? But we always quantify this based on volume of distribution, clearance, absorption, half-life, and oral bioavailability. Dosage amount and dosage frequency, of course, you require animal studies to come up, or dependent data. Now, everybody might think, how from a chemical compound you can predict an ADME? Yeah, this slide tells you very nicely about that. If we see here, absorption, the chemical properties of log-P, pK, stability, molecular weight, hydrogen bond, PSA, and Flex corresponds to absorption. This is what I was telling you in the SAR prediction. The relevance, mechanistic relevance. If those descriptors of chemical compound has relevance to the activity, that makes a good sound of relevance of science, not simply R square, right? So this is how the ADME properties are predicted from the 2D structures. These are the different ADME properties available from a software called a StarDrop, and it can predict hERG liability, human intestinal absorption, blood-brain barrier, plasma glycoprotein, protein binding category, and then many more others. Of course, there will be standard deviation predicted from here. Otherwise, applicability domain will not be known. Another is P450, which we talk about the regioselectivity. So this is a compound. We were looking at P450 isoforms, and we found that this particular carbon is unstable, so that it could break down. And this nitrogen is also unstable. To be honest with you, this is also unstable. So it depends. As I told you, software cannot completely predict everything. You should put your expertise too. This is what happened. When this nitrogen, from here nitrogen, there are another 2 methyl groups where that carbon was unstable, it formed a keto group. And this nitrogen as such, it is there. So this tells you about the composite site liability. Lower the value, better the stability. If it is closer to 0, it is more stable. If it is closer to 1, unstable. And now comes a direct nexus. Direct nexus is from LASA. It's a charitable organization in UK. They collect the clinical trial data from the pharma industries worldwide. So it is a knowledge base. There's no numbers coming up. They just tell argumental data. They say whether the toxicity is certain. There is a proof that the proposition is true. For example, if it is carcinogenic, we have a proof that it is true. Probable, at least one strong argument that the proposition is true and no arguments against it. Plausible, the weight of the evidence supports the proposition. So it's all argumental. That what it is trying to say is, if you put this compound and if direct nexus is predicting this region is carcinogenic, and if it is saying plausible, the weight of evidence supports it, means this region is already being evaluated through clinical trials and understood they are carcinogenic. So there we have a proof there, equivocal doubt. So it doesn't give any value. Of course, you have to do wet lab experiments. Why this is required? Because when you're applying for FDA approval, FDA requires software predicted human toxicity endpoints. And there's only two softwares does that. One is called TopCat and another is Direct Nexus. And Direct Nexus is very widely being now used rather than TopCat because of the updates and others. There are 43 toxicity endpoints available in the model, and you can predict chromosome damage, carcinogenicity, mutagenicity, genotoxicity, irritation, mitochondrial disruption, nephro, cardio, organ toxicity, and neuro, bladder, bone marrow, then reproductive, respiratory, skin sensitive, many more data. So FDA requires this data before going for clinical trial approvals. And for a chemist, as I told you, we always look at options for optimization. What softwares can give idea from property to structure prediction. That is also possible. So here, I wanted to reduce toxicity, or I want to improve some of the property. So the software was a database was able to predict lactam to pyrimidine. Now in this, in the participant, how many of you know straightforward how to make or convert lactam to pyrimidine, right? But if you still don't know, you have references being given at the bottom. So how to convert lactam to pyrimidine. And there are optimizations where you can use docking results to make modifications in your structure. Oh, sorry. And then also you can make reagents. Also, we can take it off like reactions and reagents, so R group analysis, and then bioisosteo replacement. When you replace a red colored region to the other, you will see change in activity. Pro-drug strategy, earlier I told you P450, like if it is breaking down and forming a metabolite, you can put a pro-drug strategy so that it won't break down. And this is another example for optimization. Terfenadine, the HERG, that is cardiotoxicity, is around 6.7 PIC50, but for fexofenadine, it is 5.2. It has reduced. The only difference is the below, the below carbon or methyl group is replaced with the carboxylic acid, and you can see the toxicity has reduced. And also for a modeler, now we have access to retrosynthetic pathway where you just give the new compound, it will tell you the possible routes, but again, based on similarity. Here it says 65% similarity. It will give you the literature references also, which will be helpful for a synthetic person, because this might be, compound might be designed by a modeler who doesn't know synthesis, but he was also able to come up with the retrosynthetic pathway. And this is what I was telling you about applicability domain, where all these green colored dots are the compounds in your training set. And this is the domain. If your compound structure or chemical class or chemotype matches with the training set, the standard deviation will be less and your prediction is more reliable. If your compound is slightly outside the applicability domain, the deviation will be less or more than the other one, more than this one, but it's kind of okay prediction. But if your compound chemical class or chemotype is very good, very big difference, like similarity is very less, then even though it predicts the standard deviation is more, the results are unreliable. That doesn't mean that the software is bad. That does mean that your chemotype of compounds are not present in the training set where the software is developed. So it is very important for any ADME predicting tool, what is the kind of training set they have prepared, what is the accuracy level of the training set that they have, and what is the predictive capability for us to understand. So many of them ask me, sir, I have used Swiss ADME, PKCSM, and many more other softwares. Which one should I consider? All are giving different data. Of course, it will give different data. In order to answer that question, try to and see whether your set of molecule is which present in which training set. If it is a paid software, if they are not disclosing, then there is some phishing, because you should ensure that the training set should be known to the user. It cannot be made confidential. How can you say that? Because that's how we believe this prediction is good or bad. So even if it is paid or free online, you should understand what is the training set is being used in order to ensure which prediction is better. If I say Swiss ADME is best I have used for my project, of course, for my structure, it worked well. Doesn't mean that for your structure, Swiss ADME is best. So you have to try all and come up to a conclusion. And this is a case study where I wanted to tell you, which I was talking about the selection in my few slides back, diversity and quality. Since short of time, let me go to the final slide. Yeah. So here we started with 3,100 compounds. We did a prediction of ADME profiling. We scored, we did a multi-parameter optimization of oral absorption and central nervous system penetration. And then we selected only 10% of 3,100 compounds. But how did we select? We gave importance to the score 25% and diversity 75%. Means if I had 100 molecules, I selected 25 molecules based upon score and 75 molecules based on diversity. I hope all chemists will agree to me, because if I just chosen only based on score, if everything is failing, it is bad. The process has gone bad. If I had diversity, some properties might be having very good activity or as expected, some might be not. But as a chemist, I can always optimize it. So only when you're considering a diversity criteria, you will be having a chance of optimization. So then we do measure in vitro potency. Then we score for the balance of the ADME and potency for those 10% of the compounds. And at the end, we give more priority to score and less to the diversity, because now we are going closer to the final narrowing down funnel. So we now give, for example, on 100 compounds, 75 compounds will be based upon the score, because we have in vitro potency data now for those 10%, right? And then 25% on the score. So this is how you have to do selection procedure. Just clicking a button, sorting them first rank and second rank doesn't work. And I hope most of you will agree here, whoever are combining computational with wet lab studies. You don't get as expected what prediction is being done on dry lab when it converts to wet lab. And finally, if you wanted to design new molecules from building blocks, this is the next era coming up. Last month, the NIH also made a very good workshop and a conference on this large fragment databases. We have a lot of databases. The best available one is NMIN database. NMIN has lots of building blocks. You can give pharmacophore criteria and others, and so that it will build up the molecule by itself, and then they can synthesize and give you. We have been working on these algorithms, and it does not do a structure similarity, but it does a pharmacophore similarity. So it tries to match the pharmacophore. For example, this particular pharmacophore is matched with this one. You can see structurally they are not similar, they are different. Right? So this is how the query and the hit looks like. The structure is different, but the pharmacophore is similar. So we do have a lot of fragments, and using the fragment space, we generate with the compounds, the reactions are already defined, and the search algorithms joins different fragments. So this is what I was telling you. So this region is being pharmacophores mapped with this region, and you can see the similarity is 0.61, local similarity. But when we combined all together, the global similarity is 0.80. So things are changing. This approach is there since 1990s. But now only people are finding its important. So imagine, that's what I said. 2011, 2010 docking is not what we are talking about 2021. So even though this technology is old, but now it is being used and applied in a different way. And we can also emphasize whether this particular region of the pharmacophore should be given more importance or less importance. And this is being already tested in Merck. I have taken a slide with their permission from Curious 2018, in 2018 I'm talking about. So they applied this particular approach in 12 drug discovery projects with 1 to 4 experiments, each one of them. Per experiment, 4 to 60 compounds, they got 80% feasibility. Means 60 compounds when they designed, around 80% of the 60 compounds was synthesizable. These are computer designed compounds using NME's data. So we are already using synthetic data and then we are generating it. Much speeder and patentable. That's a good part. So whatever designed using the computers, and if it's synthesizable and tested, it is highly patentable. So that is also promising. And there are many other tools, these 9 explanations are also in my YouTube. I'm not going to spend that time here. You can automate multiple steps together in a single click. Of course, be very careful. There are a lot of chances you can make mistakes. And also different docking modes and different file formats are also required for all these tools. So I know you will be using ChemDraw, ChemSketch, or even Marvin. So you can prepare all these file formats using any of these tools. So mole file, this is how it looks like. And mol2 is how it looks like with the hydrogen. So mol file can contain only one structure in a single file. Mol2 can contain more than one structure in a single file. SDF is something similar to Excel sheet. It can contain more than one structure as well as its data, alpha or numerical data as an Excel sheet, similar to Excel sheet. Then PDB is a protein data bank, mostly for proteins. SKC, CDX, MRV, it's all proprietary data. It is not human readable. And SMILES is what I have shown here. It's 4 carbons. So that's all from me for today. I thought if there was time, I could show you some demo, but it seems I'm already covering the time. It's already 10:30. So we do provide softwares for formulations, then co-crystal screening, solubility predictions, then ADME, toxicity, docking, fragment-based drug designing, protein modeling, molecular dynamics, and many more. And we do take interns, research interns, or only basically MPharms. We do have students from Manipal, JSS, and many more other pharmacy institutes. And they do have a project as what I discussed, same. They do in-silico work, optimize, they do synthesize, they do activity studies, and finally come up with a very good publications, not just project submissions. So we do have students, and we do take